All right, good afternoon, and I'm Piero Carnici from Human Technopole and RIKEN in Japan, and I'm pleased to chair the Technology Showcase Part 3. So we don't have so much time, so I ask all the speakers to really stay on time. If there is extra time, we can allow questions, and if there is no time, we will not, okay? But I really appreciate it. So we will have space for the next session and for everybody to speak. So the first speaker is Patrick Bogard from Roche. He's going to talk about a very interesting, introducing the sequencing by expansion as SBX. So Patrick. And I will stand here at the end. Perfect.
Awesome, thank you, Piero. Could we pull the slides up? So while they're pulling up, as Piero said, Patrick Bogard, I'm the lifecycle leader for sequencing applications ecosystems, and really it's a role where we're developing relationships in places Roche Diagnostics hasn't played before. And so we're really excited to be at our first HCA meeting. Like I kind of allude to, Roche is focused on diagnostics, and it's exciting here to see where the basic research is being done and where it's going to turn into the translational tools. And so please come talk to us. We're really interested in the ideas you have, and we're extremely excited to introduce sequencing by expansion. It's a novel technology for sequencing. And so let me walk you through it. Let me highlight some of the data that we've generated thus far. It's really going to be a teaser, and I'll share some links where you can get some more information. But please stop by the booth where there's a couple of us there, and we would love to chat. So beginning about February, we've started to unveil this technology starting at AGBT. And so there's a webinar where Mark Kokoris, the inventor of the SBX technology, has introduced it, really gone into the details of the chemistry. He put out a seminal preprint on bioRxiv a few weeks ago. We have some copies here, but please check it out. It really dives into the hardcore organic and physical chemistry that's gone into building the Expandable technology. And then we've begun an early access program, and our first two partners are the Hartwig Foundation in the Netherlands and then the Broad Institute. And just about 2 weeks ago, right before HCL, we announced a long-term collaboration with Broad Clinical Labs, really focused on two main topics in the beginning. The first is on fast NICU and really fast whole genome sequencing to give answers in rapid time using a system that can be brought to clinical use quickly. And then another one focused on single cell and really pushing the limits of our SBX technology with different single cell and spatial technologies as we go forward. So what are the benefits of SBX and what has gone into the design of it? And Mark and the team really focused on flexibility. It's a system where you're going to be able to tailor things to your needs, but you're going to be able to then span maybe going from a 10-minute run where you want something quick or fast to a 4-hour run where you need to generate amounts of data that you aren't able to in that time span. With that, we're able to deliver Q40 accuracy using our duplex mode. This is on the 7 GAB samples, and happy to share some of that data. We've seen improvements from February through March, through April, and then as we go towards launch and we lock the system, we're going to see these improvements start to slow in the launch, but we're always working on improving the technology, and we see there's a ton of headroom in this technology, and I'll share some of that. When we talk about what throughput is possible with the SBX technology, we're generating about 500 million bases of sequencing data per second. And what this leads to is in a single 1-hour run, you're able to generate 7 whole genomes at 30x coverage. And so in a 4-hour run, which is where the maximum we'll be doing on one chip, you're doing 24 to 28 whole genomes at this coverage. And so this is immense amount of data in a fast time. Now the other part is read length flexibility. We're launching with 2 different modes of library preps, and I'll go into a little more detail later. One is simplex, where the read lengths you get can be as short as the DNA you put in, because you're converting it to this expandable molecule, or up to 1,000 kb plus. And we're continuing to push what that end read length is, and I'll share some recent data where we're seeing average read lengths above 1,000 plus. And then we have our high-quality SBX duplex, which has an insert size of about 150 to 350 base pairs, where we're generating that Q40 accuracy. And this is using a hairpin adapter, which I'll show you a little bit. Kind of as I alluded to with our collaboration with the Broad, the technology is really capable in this urgent sample space. So recently the team has demonstrated going from sample to a VCF file, which has the variant calls, in under 5 hours. And so we're sequencing, in this case, a trio, so it could be a mom, a dad, and a baby at that point. And so this speed, we think, is really going to change the game in some of these pediatric NICU use cases. And then we also have a reusable sensor module. And what this is going to allow to do, and I'll talk about this later, is it really gives a lever on cost efficiency, because you have this CMOS, this sensor chip that you're using, and now you don't have to throw it out after every run. You're able to regenerate it and do the next run. And so this gives that flexibility where you're not penalized for doing that 10-minute run, because the next run you do could be 4 hours. Or maybe you're going to do multiple 4-hour runs in a row, and it's the same effect on the chip. And so how did we get to this technology? It's really a marriage of two acquisitions that Roche has done over the past 12 to 15 years. The first was Genia, which lays the foundation for our sensor. This is at this point, it's an 8 million well chip, where we have 8 million sensors that we're reading from, and that's how we're able to generate that 500 million bases per second. And then we combined it with the Spatial Genomics expandomer technology, and this came from Mark, and he's worked on it for the past 15 years as well. Independently, I don't think we'd have this SBX as we have it today, but it's really the power of combining the force of two and having the resolve to spend 10 years bringing this to a product and eventually to market in 2026. And so what is the sequencing system composed of? It's actually two parts. We have a synthesis instrument where you generate the expandamers. And so this is where you take your DNA library that you've prepared through normal library processing steps, you put into the synthesis instrument, and you convert that to this expandamer, to this very large molecule. You can do up to 4 libraries at a time, in the library pools of library, let's say, and that process takes about 4 hours. You then can load 1, 2, 3, or 4 onto your sequencing instrument, and then set your run times for those runs, and they'll just run in series. So if you load 4 on there, it's walk away for about 16 hours, and it'll just process them as they go through, regenerate the chip each time. And then we just announced last week at our DIA Day that this system will be termed Excellios. And so this will be we will stop seeing the Roche sequencing system, and we'll start to see Excellios as we go forward. And so sequencing by expansion is this transformation or translation of the DNA code into expandamer molecules. What we do is we have for every base, there's a matching expandamer. We have a expandamer synthase, which then takes your single-stranded DNA, incorporates each one of the XNTPs, so the expandamer nucleotides, and then continues until it runs out. So let's say 1,000 KB, or 1 KB, 1,000 base pairs. Each expandamer increases your signal to noise by about 50x. And so what this allows us to do is run these molecules, these expandamer molecules, through the pores at extremely fast rates. But because there's such a differentiation between the bases, we're able to get very clear base calling between each one of these. And then you can see there's a little orange triangle on each one of those. That's our translocation control element. And so built into the measurements, we actually take multiple measurements of each expandamer to increase our confidence of the base reading we're doing there. And so that we're able to control that expandamer molecule, that A, that T, that C, or the G, going through the pore. And so how does this work in general? What is the workflow? So you would load the chip onto the instrument. It flows on your pores, your lipid bilayer. You insert your pores into the bilayers, and then the next step is you flow on your expandamer library that you've created on the expandamer synthesis instrument. It sequences for as long as it's set up, so let's say 10 minutes to 4 hours. You then run a cleaning protocol, which is actually built into the protocols it's running, so that's counted into that 4-hour time. And then your chip is ready for the next run. And so all you do for the next one, the system flows on new lipid, new bilayers, and it repeats the cycle. As I stated a few slides ago, we have about 8 million wells on the chip, and regularly we're seeing 90 to 95% loading of those chips. And so you see You can see here we started with 7.3 million active pores, and then after 4 hours of sequencing, we were down to about 5 million. So there are drop-offs. Lipids pop, pores get clogged, but still you're generating so much data that even with that drop-off, it doesn't affect the total amounts that you're getting out. So just a little bit of detail on the library preps. So SBX duplex is our, again, high quality, high accuracy Q40 plus now. It contains a hairpin on one side and then a Y adapter on the other. And what this allows you to do is read through both strands and come up with a consensus accuracy where both bases are present on the parent strands. Use cases we see for this: whole exome, whole genome, somatic oncology, rapid WGS, FFP, and very specific use cases like minimal residual disease detection. Using the same sequencer and most of the same library prep, we're able to do simplex as well. And really here we just have two ends on it. We're working to get as close to the sample as possible, right? And so you'll either put on adapters that have the right sequences for the expandomer synthesis, or maybe there's ways we can take workflows that exist currently and just modify them so that they work with the SBX system. We think this is a really killer for single cell applications. The amount of data you're generating coming out of this is, in the time throughput that you're getting, it is just incredible. And this flexibility of having shorter reads, let's say less than 500, all the way up to 1 KB plus. We think bringing this to the field is going to allow you guys to do some cool science, and that's what we're looking for, is what can you use this for? How are you going to do it, and what does it open up? So just quickly on XBXS or simplex, two examples here using, coming from our collaborator at the Broad, Aziz Al Khalifa. One using 10x flex probe and one using 10x 5' MRA. And we just want to show what are the throughputs we're getting in 1 hour of sequencing. And so on the flex probe, you can see you get one clear peak at 168 bases. And in this 1 hour sequencing, you're getting nearly 14 billion reads, 13.7 billion reads. And these are high confident reads that go into Cell Ranger. So the actual amount of reads is more than 13.7. It's probably 15 to 20 billion. I don't have the exact number. But we're extremely conservative in what we call going forward as high quality, right? We're taking things that were made for sequencing by synthesis and porting them over into sequencing by expansion. And they're different use cases. And so as we go forward and the field develops and gets used to using SBX, we're going to see these numbers continue to increase. If we look at 5' mRNA, in 1 hour we got about 4 billion reads with an average read length of 530 bases. 4 billion reads with 530, and then greater than 1 KB was about 0.2 billion. And so this is where we were at in February, and the team went back and said, hey, we see a lot of utility in the reads greater than 1 KB. We're getting a lot of feedback from our collaborators at the Broad that there's some cool science there. And so we spent the next few months looking at how can we increase those 1 KB reads, and could we come up with a workflow for that? And so the team has built this workflow very similar to what we're doing, and I apologize for not showing the first one, but we really rely on this linear amplification step. We find that really drives yields of these longer molecules. And so when we do that, we're seeing data here where we're getting, average read length about 980 bases, over 1 billion reads in an hour. And so this is data that isn't really accessible using other modalities. And we think single cell is a great place for this. Maybe you're looking at isoforms, maybe you're looking at long, you're interested in those long molecules. But also as we look at whole genome, combining SBX simplex long with the duplex, you're going to start to bring changes to phasing. And I encourage everybody, if you're interested in that, to go look at the ESHG seminar we had. We have some data on that showing what happens when you combine these two modalities into one, and what are you able to pull out. So just talking a little bit about the analysis, and I will admit I am not an expert in this, so please, if you have questions, ask them, and then I can take them back to the team. But because our reads are very long and in one direction, we're not doing paired end, we don't have a read 1, read 2. We've done a lot of work on the informatics side to split the SBX reads into a read 1 and read 2 FASTQ, and then port those right into Cell Ranger. So all the work that Aziz and the collaborators at Broad have shown, we're taking our reads, we're making them usable with the existing tools, and then porting everything out so that you're then able to put it back into your own pipelines that you've made. This is extremely important with us, that as we come into the sequencing ecosystem, The tools that have been developed may need to be modified slightly here or there, but the data that's coming off of our system goes right into it and enables you to keep doing amazing work. I just want to talk about accuracy over length. And so here we have 3 different templates, one about 400 base pairs, one 500, one 750, and one 1 KB. We do not see a drop-off in quality over sequencing. And this is because you have your expander molecule and it's just reading through, and it's the pore is just reading the voltage change basically. And here you can see we're at Q21 across the board, even at long reads. And so this is, you're not penalized by doing longer, and there's no penalty by including a 1 KB with a 350 base pair fragment. You're able to put it all together and sequence. And then one last thing I just want to touch on is just our analysis pipeline. And so the team has spent a ton of time figuring out how we deal with the amounts of data that are coming off of this. And so working with collaborators at companies like NVIDIA, we have taken our primary and post-primary processing and made it all online. And so for a single molecule that you've sequenced, that single molecule goes through base calling, it goes through demux, intramolecular consensus, and very shortly, mapping and alignment without being written to memory. It's only after you've mapped it is it written down. So we're saving tons of data space here, but we've built into it different parts where you can pull it off depending on your use case. So if you want to pull it off after demux and get your simplex read files, that's the place. If you want to pull it off after intramolecular consensus for duplex, you can do that. Or if you want your BAMs and CRAMs to come off of the compute, that's also possible. And we're building a suite of open source tools that will be available at launch as well to work with the data types that we're coming off. And so, just want to leave you with the flexibility the system has. Hopefully it piques some ideas in your head and say, how could I use something like this? How does it change how I'm thinking about doing my sequencing now? And we will be in the booth and excited to talk to you guys. So thank you.
All right, Patrick, thank you very much. It was wonderful technology, and I think that we are at the end of the planned 15 minutes. And all the presentation from now will be 10 minutes. So the next speaker is Patrick Murphy from Oxford Nanopore Technology. We'll talk about advancing spatial and single-cell transcriptomics with Oxford Nanopore.
Thank you very much. Hi, everyone. So my name is Patrick Murphy. I'm the market segment manager for single cell and spatial for Oxford Nanopore Technologies. And today I think I'm going to talk about something that's probably pretty cool, or could be close to the heart of people at HCA, and that's getting more data back from your samples, your very critical and important biological samples. So the way that we would think about getting additional information back from our samples is through not just doing a short read gene counting approach of about 90 base pairs to figure out what genes are present in your sample, but actually getting the full length transcript to see what additional information is present in your transcripts. And so here, if we look at the center of the graphic, we know that poly-A capture approaches typically have a bias towards either the 3' or 5' end, depending on the chemistry. And so if we pair that to a short read approach, what we're gonna get are reads that pile up on either the 3' or the 5' end, which is great for gene counting, but does cause us to lose the information that's further down the gene body. If we compare that to a full-length read from Oxford Nanopore, what we can see is, in this example off the 10x platform, we're able to get transcripts that cover, or reads, I should say, that cover the full length of the transcript. And here we can see the different isoforms that are present in this particular gene that's shown on the screen. And so to make this a solution that can be really easy to use, we have a complete end-to-end solution. So essentially you start with really any full-length single-cell or spatial cDNA. That can then go into our library prep and sequencing, but we are a compatible product with 10x Genomics, so we have protocols that have been completely worked out on the wet lab side for 10x Genomics, and then also wet lab support, dry lab support as well for analyzing those data. So we're compatible with the 3' and the 5' assays, with NextGem and GemX technology from the Chromium side, and from the spatial side, Visium V1. And so what you get at the end after you've done your library prep and your sequencing is going to be then what you normally get from a single cell run. You get a cell by gene matrix, but you're also going to get cell by isoform matrix, cell by SNP matrix, and cell by fusion matrix. So now we can start to see that additional information that's present in our transcripts. So we're all in this room probably who've done a lot of short read, probably Illumina sequencing at this point. And so the question is, of course, how does this actually compare at the basic level of clustering, cell type identification if we're looking at short reads versus long reads? And so here on the left side of the screen, this is work done by Ray Chen, who is one of the organizers of the HCA network. His lab looked at eye samples, sequenced them with short read sequencing, and then sequenced them with Oxford Nanopore long reads. And what they found, as you can see, is very similar clustering. Same cell populations are present. So at the most basic level, if you're used to using short reads, this can port in directly to give you the same level of information. But we don't want to do the same thing we've already done, because then what is the value? And so what is actually, we're going to start getting additional information is going to be from the isoforms that are present. And so another part of his work is shown here on the right-hand side, where they're starting to look at both the number and category of isoforms that are present in the sample. And they can see tens of thousands of isoforms, about 17,000 of them which are novel. So this is completely novel information that's been identified in these samples. And then over 7,000 of them being cell class specific isoforms, right? So meaning that it actually matters that we look at this in a single cell context and not in a bulk context, because it could have some level of role in certain cell types. And what we can also do with this information is get beyond just differential gene expression into looking at differential transcript and differential isoform usage expression. So here in a different paper, on the left-hand side, we're looking at just one gene, HOOK2. We can see a couple of different transcript isoforms that are being expressed, and they're expressed in both normal and tumor cells, all of the isoforms that are seen there. But what we see, what they see in the paper is that normal cells utilize a different transcript as their most dominant transcript, and a different isoform than the tumor cells do. And so if we think about how this could play into looking at different cell states and trying to understand even further the heterogeneity of the cell populations that are present in our samples, this could be provide another layer of biological understanding to our sample. The same paper, since it wasn't it was cancer work, also took a look at what mutations they could see in cancer cells compared to normal cells. And they found, obviously, well, I would say obviously, the tumor cells most likely were showing obviously more mutations than the normal cells. And so this is something, again, that they were more able to see because they had not just 90 base pairs or so of information, but they had full 1,000, 1,500 base pairs of information from their transcripts. And so their conclusion, or one of their conclusions in their paper, was that gene expression levels may only be really revealing the tip of the iceberg of transcriptional diversity in the sample that they're studying, which is cancer. We're not limited just to single cell here. Also, this technology can be used in spatial. And so here we've got two different examples. On the right-hand side, looking at TCR expression in Visium V1. And so what this group wanted to do was see where TCRs were localizing within the tumor, but it's a 3' capture approach, and so really couldn't get that CDR3 information really reliably without sequencing the whole transcript to allow us to get both the VDJ information that's more towards the 5' end, as well as retaining the spatial barcode. And so what they did was they sequenced the gene expression using short reads, but they also took the full-length cDNA and pulled out the TCR transcripts of interest and sequenced those on the Oxford Nanopore system. And here we're able to see within the tumor that there's some localization to the TCRs that we can identify using this approach that would not be possible with short reads. On the right-hand side, we can see that this also works with the Curio-Seeker system. So this paper came out, well, its preprint came out just a couple of months ago from Cornell University in the United States. And they were interested in identifying, using spatial technology to look at kinetics of the immune response to viral infection. And so here, this is looking at day 21 post-viral infection. They were also able to use a magnetic bead pull-down approach to pull out TCR and BCR receptors, sequence them with long-read sequencing through Oxford Nanopore, and actually localize the T-cell and B-cell clones. And so we're able to then see additional information with a spatial context using long-read sequencing compared to short-read. But Mike from 10X was talking yesterday about how there's going to be new higher resolution Visium HD 3' that's going to be shipping this week. And so this is also something that's obviously quite a bit of interest to the field as well as to us at Oxford Nanopore. And so we were able to work with 10X Genomics early on to get some, they provided us with some 3' Visium HD samples to test. And here I'm showing a Visium HD 3' mouse brain sample with cDNA provided from 10X Genomics. It was sequenced on the our Oxford Nanopore's sequencing device. And because I'm not a skilled bioinformatician, I use Loop Browser to just show us, show what like counts. But in addition to this, we also have the full length information available for isoforms, SNPs, and fusions. We'll be able to process and start to tease apart more as that product comes more to launch. So at Nanopore, we like this saying, what's missing matters. And I think that really resonates with, or I hope it resonates with the HCA group, because there's, I think, a reason you guys don't use bulk RNA approaches, right? You want to see the heterogeneity that's there in your sample. And Aviv showed at the beginning, there's dozens or maybe hundreds of new cell types and cell states that have been able to be identified by this group here. But there's still a lot more information that can be gathered from the samples that you have when you're running your single cell and your spatial. If you're not looking at the full-length mRNA transcript, you could be missing complex biology, you could be missing potential discovery, and you could be missing disease targets. And so full-length sequencing from Oxford Nanopore can enable that discovery as well. And so this here is my final slide. So I just wanted to say, if you have any questions about long-read sequencing, please feel free to stop by the booth. We're right outside. Also, if you've done long-read in the past and maybe had struggles with it or had some reason to not use it, I would love to hear why, because ultimately trying to work on improving the strategy for the company and moving things forward and getting things moving towards long reads. So with that, if there's any questions, I think if there's time, happy to take questions. Otherwise, I'll be out at the booth after, or feel free to email me at any time. Thank you. Okay.
Patrick, thank you. You are perfect on time, and I really appreciate. And we have one. Thank you.
All right, thank you.
And we will visit all the booths because they have they are available and they have a fantastic products there. So the next speaker is Rakshit Chaudhary from Bruker, and he's going to talk about from pixels to pathways, transforming discovery with Bruker spatial biology.
Hello, everyone. Thank you. So I'm going to talk about expanding the limits of single cell, and I know it's not what there in the agenda, but I thought this would be a very good next step of what we are discussing and what we have been discussing in the past 1.5 days. So this is our inventor, Joe Beecham. I'm not going to talk about what this story is, but when a decade back, he started thinking about this spatial transcriptome technology or spatial omics technology, there were two things very clear to him. First, that it needs to be unlimited multiomics, and second, it should work on hospital-grade FFPE samples. So in 2019, we came up with the Genomics platform, which is a spatial profiler, and we released 18,000 gene whole transcriptome in 2019. Fast forward to 2025 now, and on CosMx spatial molecular imager, which is a single cell subcellular imager, we are now doing whole transcriptome of 19,000 genes, human genes. And we are also in process of doing 1,100 proteins, proteome atlas. So how does this all start and how we are able to do this? Our technology was very simplistic. We used direct hybridization because we knew amplification won't work on FFPE samples. Plus, it's not going to be scalable. So 18 years back when we launched Encounter technology, it was very simple. We needed 50 nucleotides and we needed a barcode. And we have just expanded that into our spatial portfolio. And that's how we do 19,000 genes now. So diving right into it, Bruker Spatial Biology, it provides end-to-end solution for your discoveries, for your translational research. And I'm going to take you on a short tour of how we do it. Talking about CosMx SMI, Spatial Molecular Imager, which is our single-cell subcellular resolution imager. The crown jewel, the latest innovation that we have recently presented during AGBT and AACR, the whole transcriptome assay. Now this is protein coding whole transcriptome of human, where we did 2.7 million cells, and we are averaging around 1,550 transcripts per cell. Nobody has even touched this 4-digit number right now. Now, I'm not going to go into the detail of how we did that. You can scan the barcode and read this preprint. We'll have the paper soon. But with this much amount of whole transcriptome imaging data, the way you see biology now changes. The way you think, way you analyze it, everything changes. You have these real pathways from all the data points of the whole transcriptome. You can have these without any inferences. You can have these without any complicated algorithms because these are the real data points from the tissue. We have these 2,700 plus pathway mapped onto the tissue one after the other. So the way we were doing cell typing, why we needed this was majorly because of the fact that we had to infer from few of the cell types, how would the whole pathway look? Now we need not do that anymore because we have the real information. We have the complete and direct biology from the tissue. And now what can we do with this biology? So this is an example of a breast cancer sample where you can actually take all these pathways and plot them in a domain. And all these domains have biological meaning. And it's fascinating how all these pathways come together to talk science. If you see the STAN domain, this is an invasive tumor. Now I want to see what is happening in the background. The light blue domain is the stress response to nutrient deprivation. I get all this information at the single cell subcellular level. Now I want to dig deeper. I want to see what is happening exactly in this invasive tumor at the cell level. How is it changing the pathway biology? Now, if you see these black dots, these T cells are bleeding into the interior stroma from the external one. How do I know what is happening there? I know this because I have mapped every single cell. I know this metabolic pathway is off the chart. It's going crazy. Whereas at the same time, what tumor is doing, it is turning off the defense mechanism, where if you see these two highlighted genes which are depleted, you won't find them in any of the existing panels which caters to FFP. Now, how do you ask a question for which you do not even know what do you find out? So this is real discovery. This is how you see what is coming up. This is the next level. So why we did this, what we did was we did direct and complete projection of all the biological pathways with best-in-class sensitivity, and we designed it for FFP. And why we did it was to get the disruptive discoveries and give you the molecular fingerprints of all these patient samples. Now, the question or the thought that I have or we have in this is, it's a very good first step for single cell. Now we have already known who they are. It's time to know where they are and what they do. We do understand with this kind of data, there's going to be a lot of challenge in terms of analysis. How do we cater that? So we have come up with something called as Atomics, which is a special informatics platform. It's very intuitive. It's multi-sample interactive analysis suite where you can do your cell segmentation. We are already in the 5th generation of cell segmentation. You can download the open source portable output formats. And just a small testimony of what it is actually doing is, you see we have already 2 billion single cell in this atomics from the users across the globe. That's more than 600 billion transcripts. And this is all ML and AI ready. So this is where we are in terms of having the data and making sense and science out of it. It's just not the end. Out of this, we are also developing a community where we have put a scratch space page. What we have done is we have a dedicated open source community for all the users where these open source softwares are available in detail. You can download your data sets, you can analyze, you can play around with them, you can do tertiary analysis and further analysis with those. Now, taking a step back in terms of resolution, we also, when we launched Profiler, we wanted to see what is happening in tissue microenvironment. And we launched in 2019 the whole transcriptome for the Profiler, and 2023 we launched the IO proteome atlas. This was for 570 proteins, and you saw a couple of examples from Dr. Arutha Kolasenge's lab on the head and neck cancer yesterday and on the 100-year-old Spanish flu cases. Now, we were expanding to whole transcriptome on the RNA level, so we wanted to make sure that we are doing equivalent justice on the proteome side. So we have come up with a 1250 plex proteome panel, which will be launched later this year. And this is 1100 unique genes. So far, the biggest proteome panel. So what are we doing with this? So I'll just give you one particular example here. We worked with Dr. Miranda Orr from Washington University, and we ran one single experiment, which yielded 15 times more dysregulated proteins on the human Alzheimer brain. Now, again, this is what we call a discovery. Now, we have all this data, and what do we do with this? How do we take it back to clinic? This is how we close the whole loop. This is how we close the whole cycle with another platform from Bruker Spatial Biology called Cellscape. This is a spatial proteomics platform which will take you from discovery to translational research. And for that, there are two very important things. First is agility, and second is customization. And this platform does both. I'll show you shortly how it does. So all it does is you prep your sample, you stain, you image them, and you erase the barcodes. This we do with a proprietary technology called EPIC IF, which just without touching or bleaching or being harsh to the tissue, it just cleaves off these fluorophores. So you can use your tissue a week later, a month later, or in some cases, a year later if you have put it in the right temperature. This added with the high resolution that this platform has, 182 nanometer per pixel, plus the HDR technology where we put low abundance and high abundance expressors on the same log scale, and the real estate of 16 into 44 millimeter square, which is the biggest imaging slide. You get a platform where you can actually convert this 30-plex protein information into a panel in just less than a month. That is the power of translational research. That is how powerful this tool is. In the interest of time, I'll just showcase you one last thing. This is something that we are coming up by end of year or early next year. This is going to be, again, groundbreaking Painscape platform, which will have 3D visualization of the genome. So this is going to be a genomic spatial platform. Thank you very much. I'll be at booth number 13 for any long discussions. If there are any questions, I can later on now.
Thank you, Rakshit, and I think that question probably at your booth. Thank you very much. So the next speaker is Jens Durruthy, and from Element, high-dimensional biology, advancing single cell, and spatial multiomics with AVT24TM.
All righty, thank you so much, Piero. It's good to be back here and to talk about our platform, the BD24. I'm the Director of Product Management at Element, and I think it's fair to say that this audience understands that biology is incredibly complex. I think Ed mentioned this yesterday in the morning session and illustrated this nicely, where he showed a mouse brain sample in the UMAP space with more than 5,000 clusters, and that's just the brain. Now, the functional unit of biology, the single cell, is defined by an orchestration between the molecular and the cellular features, both within the spatial as well as the temporal space. And so we at Element believe in order to unravel that complexity, we need solutions that have the ability to both scale at high resolution as well as at low cost, and really provide workflows that both are accessible as well as easy to use. The BD24 was really built from the ground up to deliver on these needs. It is the first single-cell spatial multiomics and sequencing solution that's fully integrated, and it enables high-dimensional profiling across a range of different applications. On the one end, you have a best-in-class, high-quality sequencer with Q50 scores, flexible throughput, and low costs as low as 70 cents per million reads. And on the other hand, you have a benchtop instrument that's essentially a toolbox that allows you to profile millions of cells across a large imageable area within the multimodal space in a very quick turnaround time. Now, with the last 20 years, NGS has really fueled multiomics tools. Many of you which have used those, ranging from single-cell spatial all the way to proteomics, and really have furthered our understanding of complex disease, such as cancer. Despite the progress that we have made, there's still a number of challenges that remain with these technologies, one of which is that these do come with lengthy workflows, often spanning across multiple days. You do often need specific and additional equipment to do from the entire workflow from start to finish, including sequencing, and you're often faced with multiple measurements that are sometimes hard or almost impossible to integrate. Now, the BD24 essentially is what I call sort of like the holy grail of the biological tool space, which is it allows you to go from sample to data within 24 hours without any library prep. And so what you have is you have a platform that's compatible with different sample types, including adherent cells, suspension cells, co-cultures, will be compatible with fresh frozen and FFPE fixed tissue as well as organoids. And you apply this to a flow cell, which you can then interrogate with certain drugs, with different genetic manipulations, and you're able to load the flow cell into the instrument and then walk away to run within 24 hours. You can do primary analysis directly on the instrument, which includes the segmentation. You can assign protein as well as RNA reads directly into the cell. And then you can do secondary analysis off the instrument, which includes aligning the reads that you get directly to reference. You can visualize it with our Canvas solution, and you can also integrate it into third-party bioinformatics tools. Now, what do you get out of this? You get essentially a multimodal view of your sample by co-detecting both RNA, protein, phosphoprotein, and 6 different cell paint markers, which characterize the morphology of your sample, all within the spatial and subcellular space. And you can imagine this to be within a fairly highly multiplexed manner. So you can add a time component to this, where you take multiple time points. The flow cells that I showed you earlier come in very flexible throughput configurations, so as little as 1 sample all the way up to 96 samples or conditions. And you can really do this within the multiomics space in less than 24 hours. And so this is just one example, what it looks like. We have Teton chemistry, which comes with multiple different panels, where you can measure both RNA transcripts up to 350, the 6 cell paint markers, which stain your organelles, as well as up to 88 custom proteins, which add to the 50 proteins that come with each of the panels. Now, just last month, we expanded on our Teton chemistry and launched what we call the Teton Atlas chemistry. This Teton Atlas chemistry measures analytes in a completely different way, and that is what I would consider a game changer. It is, there's no library required. You can expand in situ and scale beyond millions of cells in a large imageable area to up to 20 centimeters square in a single run. And you can do this across a different number of cells, sample types, including cells as well as tissue. We call this new technology direct in-sample sequencing, or DIS for short. And DIS is really sequencing the native transcript within your cell where the transcript actually occurs. It's going to do this both across the whole transcriptome, so 3', as well as in a very targeted manner. And you can combine that direct readout with our Teton barcode chemistry, which allows you to overlay protein, phosphoprotein, and cell paint markers. Now, how do we do this? There's two different approaches on how we actually get to do the DIS workflow. One of which is a set of oligos that are binding to the poly A tail to your 3' transcript. And the other one, which is an oligo that you design through your vendor of choice, IDT or Twist, and then have it bound to a sequence of interest, which then sequences downstream anything that you want, whether it's guide RNA protospacers, whether it's SNPs or express SNPs, whether it's fusion genes, anything that you think is important for your application. Downstream of that, the same process occurs, which is it extends into a single-stranded cDNA through reverse transcription. You circularize the cDNA. We then amplify that through rolling circular amplification. And then we create these clusters, or what we call pollinase element, which essentially is the template for the ABC chemistry to then read the transcript cycle by cycle. Now, there's many applications, and I would argue that although we have thought of many of them in-house, there's likely going to be a number of applications that customers will come up with once this product launches. What you see here is just 3 examples. Each of these videos reflects sort of the different pollinase or the clusters that you see within the spatial context, and as each of these pollinase is being read through the sequencing cycles. And so you can sequence up to 20 base pairs in length, which is typically sufficient for protospacers on the guide RNA. You can sequence up to 100 base pairs if you want to do some targeted RNA approaches and look at some specific SNPs or fusion features of the transcript. But you can also just sequence 50 base pairs, which is typically sufficient for you to go through the 3' end of the transcript to line it back to a reference to then do traditional gene counting. What we've also done in-house is kind of show the practicality of reusing DIS to do single-cell 3' gene expression analysis. And what you see here is taking 4 cell lines together, mixing them, and then putting them onto a flow cell, culture them for a day, fix them at 4% PFA, and then putting them directly into the VD system. And then within 24 hours, what you see is the post-analysis, the clear distinction of these cell types, which you would expect based on the 3' gene expression differences. But the beauty of this is that you can actually do this at massive scale. So this was done with 2 million cells in a given run. You can now overlay the protein as well as the cellular morphology features, which come with each and every one of these different data points, because they can trace back to the spatial context. And it's all done without any library prep, as well as no encapsulation. So you're essentially taking the sample as it comes and then putting it straight onto the flow cell. This last application is kind of highlighting the power that you can achieve with targeted sequencing. This is optical pooled screens. And what you typically do in these optical pooled screens is you take a pooled CRISPR library and you apply it to your cell line of interest, and then ask the question, how does my morphology in an individual cell change as a result of a gene that's being perturbed within a single cell? And historically, the way you've done this is kind of like looking at a few cell paint markers, 3 to 4, and you do it at a fairly high throughput, which is maybe the low hundreds of guide RNAs, but you're always challenged with the sensitivity as well as the specificity, since the readout is through barcodes as well as through an in situ readout. What we have done here in collaboration with Genentech is we essentially took that approach to the AVD platform and then applied that DIS workflow, which sequences your guide directly, but it also is able to now measure all the features that come with each of the cells, which is the cell paint markers, the protein markers, as well as the RNA markers, and shortening the workflow from a two-week workflow all the way up to a 24-hour workflow. And there's going to be a paper coming out of this. There's a poster as well, but I'm just going to show you this one slide where you have now all the guide RNAs, in which case here we looked at 2,000 guides targeting 500 genes, and you can ask the question, which guide RNA has which impact to all the features that you can measure. And we're looking at RNA, protein, cell paint features, where each of the cell paint markers comes with hundreds of morphological features that you can measure. And if you zoom into one of these guide RNAs like RPL8, you can then ask the question, which of these features are down or upregulated? And because you have the spatial and imaging information, you can go back and see where exactly your guide was expressed and to what extent it impacted the downstream up or down regulation of the genes. Now, I want to leave you with this slide, which is fresh off the press, something that we're right now really focusing on, which is applying the DIS workflow within the tissue context and being able to read the whole transcriptome across an imageable area that's 20 centimeters squared across the entire tissue. This is a fresh frozen liver tissue. You could see clear distinction between those individual cell types based on the marker genes. And we're super excited that this is going to be coming out early next year. And we're also super excited about the roadmap as a whole, which is a number of extensions on content that will come out later this summer. We are going to release the targeted approach of DIS. We're also going to release the whole transcriptome approach for the DIS method later this year. And then, as I said, I'm really excited about the tissue capability, both in fresh frozen and FFPE, that will come out in H1 of next year. Two last slides, which are quick. I do want to just highlight that we have launched a program. If you are interested in actually getting your hands on early on this DIS workflow, please submit your abstract. We do run these projects in-house and can really tailor it to your needs on your experimental design. And then if you are interested in learning more, definitely visit our booth. For those that haven't, we do have specific HCA promotions that extend beyond your HCA discounts that you currently receive. And we're really excited to think about the applications that you can run. And with that, I'm going to thank you for your attention, and I believe we're out of time.
Thank you, Jens, and fantastic work you too. So visit him at the Element booth. And the next speaker is Zohar Shipony from Ultima Genomics and driving large-scale single-cell studies with transformative sequencing economics.
Hi, everyone. I'm Zohar. I'm from Ultima Genomics, and today I'm going to tell you how Ultima is driving large-scale single-cell studies with its transformative sequencing economics. So a bit of overview of Ultima. We have been founded in 2016, so we've been around for quite some time, but we only came out of stealth at 2022 at AGBT when we announced the $100 genome, which back then was a huge price difference or where we were at about $500, $600 per genome, and we announced the $100. And this year at AGBT, we announced the $80 genome. And our roadmap is to keep driving the cost down as we go into the future. So, as I said, $80 genome announced at AGBT. If we put it in the unit of 1 million reads, the 24 cents for 1 million reads, and in the unit of a single cell at 20,000 reads per cell, it's less than half a cent per cell. And when we made the announcements, we announced the UG100 Solaris chemistry, and it took us from 6 to 8 billion reads on a wafer to the 10 to 12 billion reads today we are doing on a single wafer. That will get you about 30,000 genomes per year. As I said, the price is 24 cents per million reads. And we have done all of this while keeping our amazing SNVQ 60 quality using the PPM-seq library prep and having a really amazing DNA to sequencing yield, getting up to 30x of WGS from only 2 nanogram of cfDNA. Also at AGBT, we announced the boost mode, giving you up to 100 billion reads in a single day using short library preps, such as the 10x Flex. And with 100 billion reads per day, we can actually get up to 5 million cells in a single day. Over the last year, we've also expanded our global network of service providers. Back in 2022, people told us, where can we sequence? And we didn't have a lot of options. But today, we have Novogene, Somogene, Eurofins coming very soon, Macrogen, University of Minnesota, Broad, and many others coming in the futures. And we are also keeping expanding on our open ecosystem, growing our partnerships. We're working, of course, with all the major single-cell companies, 10x, PowerScale, BioScrub for single-cell whole genome, and many more. So if you have a single-cell library, switching to the UG100 is very simple. You simply do your favorite library prep, we do a simple PCR conversion, taking your existing library and adding our UG barcodes. Then we sequence on our on our Ultima machine, and it goes seamlessly into your third-party data software. There is no need to do any changes. Over the last year, we've also done a full compatibility test with all the major providers. On the right, you can see the 10x GEMX Flex, perfect correlation plot, and you can download the application note down at the QR. With parse, you can see the WT Mega, also a sequencing saturation showing very nice compatibility. And again, the technical note is available for download, and also with scale, very nice correlation showing our full compatibility, and an up note will come soon. There will be a QR code in the near future. Over the last years, since we came out of stealth, there were many publications using the Ultima technology, going through different types of application, and I wanted to highlight two of those papers. The first paper is the SkyMet version 3 protocol coming from the Andrew AD lab. Until now, I talked mainly about single-cell RNA, but single-cell is not limited to RNA. We showed BioScribe that can do single-cell WGS, and we can also do single-cell methylation. The problem with single-cell methylation is to get to saturation for a single cell, you need about 2 to 3 million reads. So combining the new method coming from Andrew AD lab, the SkyMet version 3, with Ultima sequencing is really a perfect combination. And together with Andrew, we produced a 140,000 cell library from the human middle frontal gyrus across 4 different individuals. And once you start doing this, you can see really amazing phenomena. You can see the UMAP showing the different populations, of course. But once you start focusing into the genes, into the actual genes, you can see how gene body methylation and promoter methylation is really different between different single cells. And this is quite amazing because people were always looking at as the methylation as promoter having no methylation, gene body having higher methylation, but you can see that really different subpopulation have a different methylation pattern across the gene. And today, using SkyMet version 3, you can generate more than 300,000 cells per experiment. Another paper I would like to highlight is from the Schluss lab at the Weizmann Institute, when they've used single-cell RNA to identify myeloid malignancies. In the Single Cell Society, like when it was 2013, 2014, the first single-cell genomics conferences, many of the visionaries were talking about how single-cell RNA will accompany blood tests. You will do a blood test, you will get your cholesterol, you will get your different chemical measurements, and then you will also do single-cell RNA measurement. 10 years have passed, 12 years have passed, and it still didn't happen. And this is not because of the visionaries. The visionaries were right. There is a need for this. This is an amazing data that can be accompanied as a blood test. The problem was that 2.5 years ago, if you wanted to do one single cell test for a single individual, that was about $1,000. Today, we're looking at an order of magnitude less. So this really opens up the possibilities to start doing that. And in the Schluss lab, what they have done CD34 enriched hematopoietic stem cell. CD34 is a bone marrow marker. So if you are taking CD34 enriched cells from the blood, you're looking at the bone marrow. There are many arguments if it's exactly showing the bone marrow or not. I will leave the arguments to the immunologist. And they have taken the CD34, did enrichment, used 10x 3' sequencing, and then first created the normal atlas for age-related hematopoietic process. So they sequenced about 100 individuals and looked at the normal development and the normal age-related phenotypes. And then when they started looking at myeloid malignancies and pre-malignant state, they noticed that they can really identify all those malignancies using only a blood test. And this is really amazing because today it's done using a bone marrow aspiration. There are many kids going through bone marrow aspiration every year. There are many adults going through bone marrow aspiration. That's a really hard process. And really, just using single cell, they can find and detect the disease. And single cell really gives you an amazing, amazing look. Also at the copy number variation, where you can see copy number variation, opening it only specific clones in the blood. Over the last year, we've also engaged in many of the big projects that are coming. Doing with scale, the 100 million cell challenge, already generating the Tau 100 million cell dataset, which is the biggest dataset out there in existence, together with parse. the UK Biobank with the 600,000 samples of Olink, and the CZI 1 billion cell, and the Arc Institute massive virtual cell initiative are all done using Ultima technology. I'm sorry that I don't have a QR code because it's already done. You cannot submit your grants anymore. But in the last month, we have opened the Count on Us initiative, where we gave the community 3 trillion free reads. It was overly subscribed. There were 350 submissions coming from 188 institutes asking for 15 trillion reads. We could not supply all of them, but we will supply a nice portion of them. And part of the 350 submissions, 25% of them were single cell projects asking for more than 200 million cells to be sequenced. And I will end with this slide. And this slide, again, if I would show this 2 years ago, people would laugh me off the stage as the sizes of projects that we show here. What we show as a small project, it is certainly not a small project. It's a big project, very big project, and a huge project. Would not have happened 2 years ago. But today, again, we can make it happen. Today, together with our amazing partners, 10x Parse and Scale, we have really driven the cost of single cell down, and all of this is now available. So if you want to discuss, if you want to do such a project, come to our booth, talk to us. We can help you drive each one of those projects. Thank you very much.
All right, wonderful, Zohar, and let's visit the Ultima Genomics booth here ahead of time. If there is any question? No, let's get back some time, thank you. So the next speaker is Xiaohui Man from BD Biosciences. And she's going to talk about level up your research with comprehensive and complete single-cell multiomics solution, BD Rhapsody. Please.
Hello. It's okay. So my name is Xiaohui. I'm Market Development Manager at BD Biosciences. So today I will introduce our single-cell multiomics solution. So we have the instrument. We call this BD Rhapsody Express, which is the legacy system. And also last year we introduced this Express, which can enable capture of up to 1 million cells. And also we have this BD Rhapsody scanner, which is really very unique in the market, which can really enable you to visualize your single cell capture process. And also we have all the assays for single cell, like WTA, ATAC, CDRR, TCR, BCR. We also have single cell proteomics, and also the buffer for tissue and cell preservation. Also the bioinformatics software we just launched, which is called Salismol. It's totally free. It doesn't need any coding experience. So I will highlight the OMIS-1 protein panel we just launched for the static analysis. So static, just to keep everybody on the same page, is like to basically to simultaneously perform both protein and mRNA at the same single cell. So the advantage of static is to really enhance the cell cluster resolution, identify the new cell population, and also identify the new cell functional stage. So shown here, just a single like UMAP. You can see in the middle, the UMAP. We just add a few markers, including CD103 and a few other antibody markers. You can see the very efficiently cluster of the cell population, also clearly visualize the CD103 cluster, which is so-called memory T cell, which is normally resident in the skin. And on the left side, you can see this UMAP actually just clustered based on the mRNA profile. And we really lose the resolution identification of this CD103 memory T cells. So now also this generation of this cluster is totally driven by the protein expression of CD103. And this is the panel we just launched recently. So we have the T cell, B cell NK cell, APC, tumor panel, all these like 30 plex. It's always like the dry down panel in a single tube. All you need to do is just you add the water, then stain your cells, and load the cell into any platform for the single cell capture. And also all these panel can be combined to have this 90, 60, 150 panel. So the panel is really, really, really cost effective, yeah. So shown here is just a panel, the T cell panel for really very deep characterization of T cell populations. And also for this panel, actually we employ the idea of the SMART. SMART idea basically is to respec in some antibodies to really partially mute the signal from very highly expressed marker like CD45. And this way we will have more reads assigned to secondary and tertiary markers, which will really enhance the resolution, also reduce the sequencing risk. So slightly different topic to mention the benefits of SASIC on the open well system like BD Rhapsody system. So you can see here, we all know, right, the SASIC data quality is really highly correlated with how clean your data, also how clean your cell prep is in terms of these unbound antibody oligos. Let's say if you have really better wash, you can really have very low amount of unbound antibodies, which will give you very specific signal. Also, people call this more accurate biology. But if you don't have the better wash, you will really have very high background of the unbound antibody, which will give you very, very significant background noise and also the less accurate biology. Yeah. So here I just want to show the single cell capture process, or basically the animated version of what happens inside of the cartridge. So basically we load the cell and the cell will really settle down into this microvalve based on gravity. So this is really nice. There's really not a lot of stress at this stage. And then we load the magnetic beads. This bead actually has been coated with millions of the oligos. Then we flush out the actual beads. Then actually the main goal of this step is to partition the cell, basically to have the one well with one single cell and the one magnetic piece. Then we introduce the lysis buffer to lyse the cell, so all the analytes from cell will be released and bound to this magnetic piece. Then we use the magnet to pull all the magnetic beads into a single open top tube for the CDN synthesis and the library preparation. So basically the idea to show this, the workflow in cartridge is really to show the very significant strength of the open well system versus this closed droplet-based system in terms of the SASIC performance. So you can see like just now the protocol, the workflow, we really said there are a lot of work steps, like we load the cell, then we wash the liquid, we load the magnetics, and also we wash the magnetic beads at least twice. So all this wash will really remove a lot of unbound antibodies, which really give you a very clear background and more accurate biology. So here I just show this, the published study to compare this droplet-based system versus the microbial system in terms of the SASIC performance. So just by eye, you can see very clearly, very specific signal using this BD Rhapsody system compared with the droplet-based 10x genomics. Yeah, another two advantages I want to mention is like BD Rhapsody has been really considered as a gold standard for profiling the fragile cell, like the granular cell. All the top publication for this granular cell, you really use the BD Rhapsody system because of the gentle capture system I just introduced, and also the very high sensitivity of our WTA chemistry. So this I want to highlight the HC price for single cell capture. So our HC price basically we can capture up to 100,000 cells per lane, which is really cost effective for single cell CRISPR screen. Another benefit I want to mention is about the high sensitivity of our WTA chemistry. Basically, for single cell CRISPR screen, we don't need the actual guide RNA enrichment step. So the WTA is already capable of detecting the guide RNA, which will really minimize the workflow and also reduce the sequencing cost for guide RNA sequencing. So this is a particular study we collaborate with the scientists in Duke NIS. You can see actually he did 3 time points for single cell capture. He aimed to detect like 510 guide RNAs and want to capture 80 cells per time point. So basically, we know about 60,000 cells, and after remove the low quality cells and the doubler, we get 40,000 cells per time point, which is really good. So this is the last slide, just to highlight, we use the micro-based system for single cell capture. Our capture system is like very gentle, so we can really capture a lot of cells, like capture the fragile cells, and also we have very high cell capture rate, up to 80 to 90%, and we don't have the clogging issue associated with this doubler-based system. And also we have this CDABs, which can be stored up to 1 year, which is really good for multi-center and longitudinal studies. And the last point, we have this scanner, which is really very unique in the market. It really enables you to visualize your single cell capture. Yeah, that's all. Thank you.
Thank you very much. And please visit the BD booth. And the last speaker is Sujatha Narayanankutty. So we're going to talk about from the Singleron is going to talk about pushing boundaries, discovering signal missed by standard single-cell RNA sequencing.
I'd like to start by thanking HCA for giving us a slot to talk in this technology showcase. We're honored to partner with HCA as well to provide cutting-edge single-cell genome multiomics analysis. So we're Singleron, and I'd like you not to think of us as just another single-cell company. We're really looking to provide sample-to-answer and accessible solutions to researchers. And so a quick sort of throwback, we were founded in 2018. Our core technology developed at Yale, and we've spun off from there, and now we have our HQ in Germany. And so over the last few years, we've really built up our customer in terms of the different organizations we work with. We've got lots of labs where our technology is used. We've got real experience with many different sample types. We're part of a lot of ongoing clinical studies, and we've got more than 600 publications where researchers have used our technology. So just sort of the different areas where we have publications, we do have them in Atlas studies, so that's sort of basic, but you can also see our technology being used in the fields of immunology, developmental biology, biomarker drug development, and also want to point out that our technology is species agnostic, so we've got a lot of studies coming up on different organisms. Right, so I mentioned sample to answer workflow, and simplistically, this is kind of what a single cell workflow looks like, everybody knows. And what I want to highlight is we really contribute to every step along the way. So starting from actually preserving your sample, be it a biopsy or a sample that you've sort of collected, or you have to look at a large-scale study, we have an option to actually preserve your sample. We have options to dissociate that into a good single-cell suspension, which is sort of the basis of setting up your single-cell workflow. Kits that help you capture the mRNA and other layers of data, and finally, analyzing all that data you get from this. And so you can actually buy products from the whole workflow or choose which one you want and integrate into the other steps that you've already optimized. The other sort of offer that we have is actually implementing this end-to-end workflow in our Singleron service labs. And so that would look something like discussing your project with us. We kind of offer you all of the different options that you can look into that suits best to your research needs, and then we can pick up your sample or ship your sample in, do the whole workflow, and hand you back your data from which you can gain your insights. And so where are we based? I'm from the Singapore office. I'm Sujatha. I head the technical support, and we cover APAC along with our trusted distributors. We also have service lab in the EMEA region, so we have our office and service lab in Germany, and then we also cover America with our office and service lab in Michigan. So we really have a worldwide presence now, and you can reach out based to the one nearest you. Okay, so a quick sort of snapshot on what our technology is. We are a microwell-based technology, and the basic is the scope chip. So it's a little chip with hundreds and thousands of wells onto which you can load your cells. And as I mentioned, we're accessible, so we're an instrument-free method. But if you like your automation and one walk-away time, we also offer an instrument that you can use or integrate into your workflow. And so what can you do with our kits, or what are researchers working with us? So this is just an example of a group that works with hepatocellular carcinoma. So they're interested in the immune cells. And based on their hypothesis, they've moved this to a mouse model working on liver samples. So they've kind of taken out the liver samples and need to make a single cell suspension with high viability and good cell numbers. So they've used our tissue dissociation option here. And post that, they actually were enriching for just immune cells of interest, where they've used our kit to do single cell profiling. And so they get all this really nice data from which they've gained their insights on how they want to propose a therapeutic strategy. So those were clinical samples, but if you're working on organoid models, so we have researchers who've actually dissociated their organoids, and here they were studying a mitochondrial disease and used our kits to figure out all the different cell types, and from there looking at gene pathways of interest. Of course, that's just all of single-cell RNA sequencing, but what if you want to go a little more? If you're interested in actual immunoreceptor profiling, we do have TCR and BCR kits. So the sort of use case would look something like this, where you're actually interested in looking at the immune tumor microenvironment. And so you take your samples and run your single-cell RNA, and on top of that, you actually get your TCR repertoire or BCR repertoire of interest. And sort of the last one I'd like to highlight is if you're looking at flash frozen samples, so you actually already frozen your samples in an interesting clinical cohort study, you can still work on them. You can do your single nuclei sequencing. So here's a kind of case where they've used muscle biopsy samples to kind of understand the potential drivers of a disease. And so they kind of did single nuclei and got interesting data to kind of pinpoint the progenitors. Right, so that was kind of what you can do basic with single cell sequencing, but my talk was kind of titled, what are we missing and how you can do that? So what would be signals that you miss by just using all of these standard single cell kits? And then just twisting that to think about what could be extra dimensions you could add to your study. And these are sort of just some points that I've listed down. So you've got SNPs that could be of interest, and important gene fusions, non-polyadenylated signals. By base, we're actually usually capturing the poly-A. If you're interested more in splicing isoforms, and then any sort of rare or low expression transcripts. So at Singleron, we sort of have options, and we've kind of divided this into two kits that I'm going to focus on over the next few minutes. One is a more targeted approach. So here you kind of want to have an hypothesis or an idea of what you're working on. And so this would look at, these would be like pre-validated kits looking at single cell mutations or gene fusions. So the three that we have are for lung cancer, clonal hematopoiesis, or blood cancer panels. And then we have another set that looks at non-poly-A transcripts, so viral or bacterial. So just a little bit into these kits. So this is kind of what the marker list would look like. For the lung cancer, it's druggable mutations, whereas for the clonal hematopoiesis case, these are sort of mutations seen in the disease. And just to kind of reiterate, why do we need these sort of specific targeted kits? If you just do a poly-A capture, you can see that the detection rate is much lower than when you actually target for these genes. And so it's just an example there. And this is sort of our blood cancer panel. So here we've designed it a little differently. You have the SNP mutations, but we've also added gene fusions that play an important role in such malignancies. And sort of maybe the easy way to understand that would be a use case. So here we have a group that was interested in understanding the mechanisms of myeloid tumorigenesis, and they kind of know there's this complex interplay of all these mutations. So you can go in and do your regular RNA-seq to get your different cell types in the sample. But on top of that, they were able to sort of zoom into the different mutations or single cell mutations to the different cell types in these samples and compare them. And based on that, kind of identify pathways that are probably sort of causing resistance to chemotherapy. So here they've kind of understood the disease heterogeneity and then kind of been able to propose a novel targeted treatment because they have this extra layer of data. So kind of switching to detecting non-polyadenylated signals. So the two that I'd kind of focus on are virus and bacteria. We have an EBV kit, so this is sort of a virus that's usually seen in cancer as well. And so a sort of use case here to show how you can profile virus at the single cell level and then kind of understand the disease. So single cell giving you all the different cell types, and on top of that, you can add this sort of data, finding out which of the different cell types that are infected by EBV, and then looking at how that can correlates to the disease condition. And at the end of this study, the authors were able to sort of identify that hematopoietic stem cells are where these are originating, and sort of the treatment that you'd want to give to one of for this kind of disease state would be to actually try transplantation. We have an intracellular bacteria kit that's just been launched. So here we actually kind of detect 16S signals at the single cell level along with the RNA of the cell. Just a little bit data to show that if you're looking at oral cancer, you can actually go in and detect the cells that have any kind of bacterial presence. And then using that sort of 16X signal, you can actually detect which species, which genera it belongs to. And sort of the last one I'd kind of point out is that all of these validated kits may not be your interest. We actually can produce kits that kind of target your markers of interest. So you can just reach out to us, and this is something we can put together so you can sort of use that for your research project. So that was the targeted panel way of looking at things. We also have sort of a step back and sort of discovery-based way. And this would be to actually get full-length single-cell RNA sequencing by using short-read sequencing. So in general, more coverage of the transcript means you get more of all of these things. So why not? And so what we've kind of developed is a protocol where we can actually sample both the 3' and the 5' readouts from a transcript, eventually putting together the full length. So this is just how the technical part works. And so as expected, what we see is that we get more cells, more genes, and most importantly, more splice junctions. The main aim is to get coverage across the middle of the transcript, and we're able to get that with this kit. And we also have an option of actually, when you get more coverage, getting reliable detection of fusion genes. So you can either target capture them or use the full length kit to figure this out. And here's an example of another fusion gene that was detected. This is part of a clinical study. So just doing both the 5' or 3', you don't detect a cluster with a strong signal. But if you actually have the full length transcript, you see that there's a group of cells that have this fusion gene. Again, sensitive detection of splice sites, splice junctions. Very clearly, if you're able to do the full transcript, you have much more robust information on this. And then more effective SNP detection as well when you get longer transcript information. And sort of the last couple of slides here, just to kind of show that, of course, you also get reduced gene dropout at the single cell level, just by sampling the entire length of the transcript. And the sort of takeaway here is that using either targeted focus scope kit that we offer or a more discovery full-length kit that we offer, you can sort of add all of these extra layers of data that you could be missing by just running a standard single cell workflow. A quick snapshot of all of the kits that we offer. I've focused on two here, along with the general and immunoreceptor profiling kits, but we do have some specific ones that look into glycosylation levels on the surface and then RNA dynamics. So I've not been able to cover a lot of the other applications. Please do come talk to us. If it's either tissue dissociation or single cell workflows, I don't recall our booth number, but we're the one that's giving coffee, so please do come find us outside. Thank you.
Thank you, Sujatha. And let's visit the Singleron booth as well.